Hello everyone and welcome to our laboratory presentation. Today we will be discussing practical 7, the detection and identification of Listeria monocytogenes and related Listeria species. Foodborne pathogens pose as a severe threat to global public health and the food industry. Among these, Listeria monocytogenes is particularly notorious due to its ability to survive in harsh environmental conditions such as high salt concentrations and low refrigeration temperatures. While it is a rare infection, listeriosis carries a high hospitalization and mortality rate, particularly among vulnerable populations such as pregnant women, newborns, and immunocompromised individuals. For this reason, regulatory bodies like the FDA and USDA have established strict zero-tolerance policies and highly specific protocols for monitoring food products and processing environments. In this presentation, we will walk you through our 5-period cultural and biochemical investigation to isolate and presumptively identify this organism from our samples. Let's look at the science behind our methodology. Regulatory standard methods rely heavily on sequential cultural enrichment, followed by selective isolation and biochemical confirmation. Because bacteria in processed food are often stressed or sublethally injured, a direct plating strategy would fail. Therefore, we utilize a two-stage enrichment process. The primary enrichment utilizes a rich medium with a low level of selectivity, allowing injured cells time to repair and recover. Once recovered, the sample is introduced to a high selective secondary enrichment medium designed to outcompete background microflora such as gram-negative bacteria and enterococci. Following enrichment, differential agar media are utilized. These media exploit specific biochemical pathways, most notably the hydrolysis of esculin. Listeria species produce an enzyme that splits esculin into esculatin, which reacts with iron ions in the media to produce a distinct black precipitate, allowing clear visual differentiation from non-esculin hydrolyzing organisms.
The specific objectives of this laboratory exercise were threefold. First, to safely isolate presumptive Listeria species from food and environmental sample using a modified, simplified USDA protocol. Second, to differentiate Listeria from common background contaminants using selective differentiation media including Fraser broth, modified Oxford, and Palcam agars. And third, to characterize our isolated colonies systematically using morphological examinations, cream staining, and classic biochemical tests like catalysis and hemolysis profiling. Our experimental timeline spanned through 5 distinct laboratory periods, with the first period done by the lab assistants, closely matching standard diagnostic workflows. In period 1, 25 grams of our food sample are weighed into a sterile stomacher bag, added 2025 milliliters of modified University of Vermont or UVM1 broth and homogenize it for 2 minutes before incubating at 30 Celsius for 24 hours. In period 2, 1 milliliter of our UVM1 culture was transferred into a tube of Fraser broth and incubated at 35 Celsius for another 24 hours to execute the selective enrichment phase. Moving to period 3, we performed 3 phase 3 isolation from our enriched Fraser broth onto 2 types of selective agar, modified Oxford or MOX agar and Palcam agar alongside positive and negative control strain. In period 4, presumptive dark colonies displaying esculin hydrolysis were picked from our isolation plates and subcultured onto triptych soy agar with yeast extract or TSAYE for morphological purity and stuffed into triptych soy agar with blood to set up our hemolysis observation. Finally, in period 5, we evaluate our TSAYE plates under oblique light and perform gram staining and microscopy analysis.
Let us examine the empirical results gathered from our practical. On our screen, the left image shows our subcultured soy plate labeled Group 7 Lab 5 Listeriaceae from period 4. As you can observe, the strict isolation was highly successful, yielding isolated, uniform, round, off-white to translucent colonies in the final phase of the streak. The clear background of this non-selective medium prepared us perfectly for colony morphology assessment. On the right, you can see the glass microscope slide prepared during period 5. This slide contains our heat fix and processed bacterial smear using the standard gram staining reagents. The crystal violet iodine complex is neatly localized in the center of the slide, indicating an appropriate concentration of cellular material ready for high-power oil immersion microscopy. When observing our gram stain slide under the microscope, we evaluated multiple fields of view to ensure accurate interpretation. In our initial fields, shown in the top two quadrants of the slide, which correspond to image 3 and 4, the smear was notably thick. In these regions, the bacterial cells are heavily aggregated and tightly packed against a dark red background. This thick clumping traps the primary crystal violet stain, making individual structural boundaries difficult to resolve. However, when moving to the ideal, thinner margins of our smear shown in the bottom two quadrants corresponding to image 5 and 6, the individual cells are beautifully resolved. In these fields, the bacteria clearly retain the primary crystal violet dye, demonstrating a definitive gram-positive reaction. Furthermore, look closely at the single scattered cells. They present distinct, uniform, very short roots or cocobacillary structures. These morphological findings perfectly satisfies the standard preference criteria for Listeria species, which characteristically present as gram-positive short roots.
To confirm our result, we need to understand how the selective media work in our experiment. The chemical used in the broth and agar such as lithium chloride, nalidixic acid and acrylamine help stop the growth of other bacteria like Enterococcus faecilis. This allowed Listeria to grow more clearly. On the stayer blade, the colony appeared blue-gray under a 45-degree light angle, which matched the typical appearance of Listeria reported in reference. Under the microscope, we observe gram-positive short rods, which is an important characteristic of Listeria. To fully confirm the bacteria according to the USDA method, more tests are needed in period 5. One test is the catalysis test, where bubbles form after adding hydrogen peroxide if the result is positive. Another test is TSA blood agar step test. Listeria monocytogen usually form a small clear zone called beta hemolysis around the step line because it produces listerolysins or this helps distinguish it from other Listeria species that do not show hemolysis. In conclusion, this practical successfully showed the steps needed to isolate and identify foodborne pathogen. Through enrichment, selective plating, and microscopic observation, we isolated a bacterium with characteristics similar to the Listeria species, including gram-positive staining and short rod-shaped cell. This experiment showed the importance of classical microbiology method in food safety, public health protections, and quality control in the food industry.